1.4 Genetic Foundations. Perhaps the most remarkable property of living cells and organisms is their ability to reproduce themselves for countless generations with nearly perfect fidelity. This continuity of inherited traits implies constancy, over millions of years, in the structure of the molecules that contain the genetic information. Very few historical records of civilization, even those etched in copper or carved in stone, Fig. 1-31, have survived for 1,000 years. But there is good evidence that the genetic instructions in living organisms have remained nearly unchanged over very much longer periods. Many bacteria have nearly the same size, shape, and internal structure as bacteria that lived almost 4 billion years ago. This continuity of structure and composition is the result of continuity in the structure of the genetic material. Figure. Among the seminal discoveries in biology in the 20th century were the chemical nature and the three-dimensional structure of the genetic material, deoxyribonucleic acid, DNA. The sequence of the monomeric subunits, the nucleotides, strictly deoxyribonucleotides, as discussed below, in this linear polymer encodes the instructions for forming all other cellular components and provides a template for the production of identical DNA molecules to be distributed to progeny when a cell divides. The perpetuation of a biological species requires that its genetic information be maintained in a stable form, expressed accurately in the form of gene products, and reproduced with a minimum of errors. The effective storage, expression, and reproduction of the genetic message defines individual species, distinguishes them from one another, and assures their continuity over successive generations. Genetic continuity is vested in single DNA molecules. DNA is a long, thin, organic polymer, the rare molecule that is constructed on the atomic scale in one dimension, width, and the human scale in another, length. A molecule of DNA can be many centimeters long. A human sperm or egg, carrying the accumulated hereditary information of billions of years of evolution, transmits this inheritance in the form of DNA molecules, in which the linear sequence of covalently linked nucleotide subunits encodes the genetic message. Usually when we describe the properties of a chemical species, we describe the average behavior of a very large number of identical molecules. While it is difficult to predict the behavior of any single molecule in a collection of, say, a picomole, about 6 × 10^11 molecules of a compound, the average behavior of the molecules is predictable because so many molecules enter into the average. Cellular DNA is a remarkable exception. The DNA that is the entire genetic material of an E. coli cell is a single molecule containing 4.64 million nucleotide pairs. That single molecule must be replicated perfectly in every detail if an E. coli cell is to give rise to identical progeny by cell division. There is no room for averaging in this process. The same is true for all cells. A human sperm brings to the egg that it fertilizes just one molecule of DNA in each of its 23 different chromosomes, to combine with just one DNA molecule in each corresponding chromosome in the egg. The result of this union is highly predictable: an embryo with all of its 20,000 genes, constructed of 3 billion nucleotide pairs, intact. An amazing chemical feat. Worked Replication. Solution. 1 gen. A single. Figure 1 to 30. 2 strands. The structure of DNA allows its replication and repair with near-perfect fidelity. The capacity of living cells to preserve their genetic material and to duplicate it for the next generation results from the structural complementarity between the two strands of the DNA molecule Fig. 1 to 32. The basic unit of DNA is a linear polymer of 4 different monomeric subunits, deoxyribonucleotides, arranged in a precise linear sequence. It is this linear sequence that encodes the genetic information. 2 of these polymeric strands are twisted about each other to form the DNA double helix, in which each deoxyribonucleotide in one strand pairs specifically with a complementary deoxyribonucleotide in the opposite strand. Before a cell divides, the 2 DNA strands separate and each serves as a template for the synthesis of a new, complementary strand, generating 2 identical double helical molecules, one for each daughter cell. If either strand is damaged at any time, continuity of information is assured. By the information present in the other strand, which can act as a template for repair of the damage. The linear sequence in DNA encodes proteins with 3-dimensional structures. The information in DNA is encoded in its linear, 1-dimensional, sequence of deoxyribonucleotide subunits, but the expression of this information results in a 3-dimensional cell. This change from 1 to 3 dimensions occurs in 2 phases. A linear sequence of deoxyribonucleotides in DNA codes, through an intermediary, RNA, for the production of a protein with a corresponding linear sequence of amino acids, Fig. 1 to 33. The protein folds into a particular 3-dimensional shape, determined by its amino acid sequence and stabilized primarily by non-covalent interactions. Although the final shape of the folded protein is dictated by its amino acid sequence, the folding is aided by molecular chaperones, see Fig. 4 to 30. The precise 3-dimensional structure, or native conformation, of the protein is crucial to its function. Once in its native conformation, a protein may associate non-covalently with other macromolecules, other proteins, nucleic acids, or lipids, to form supramolecular complexes such as chromosomes, ribosomes, and membranes. The individual molecules of these complexes have specific, high-affinity binding sites for each other, and within the cell they spontaneously self-assemble into functional complexes. Although the amino acid sequences of proteins carry all necessary information for achieving the protein's native conformation, accurate folding and self-assembly also require the right cellular environment pH, ionic strength, metal ion concentrations, and so forth. Thus, DNA sequence alone is not enough to form and maintain a fully functioning cell. Figure 1 to Summary 1.4 Genetic Foundations. Genetic information is encoded in the linear sequence of 4 types of deoxyribonucleotides in DNA. The double helical DNA molecule contains an internal template for its own replication and repair. DNA molecules are extraordinarily large, with molecular weights in the millions or billions. Despite the enormous size of DNA, the sequence of its nucleotides is very precise, and the maintenance of this precise sequence over very long times is the basis for genetic continuity in organisms. The linear sequence of amino acids in a protein, which is encoded in the DNA of the gene for that protein, produces a protein's unique three-dimensional structure, a process also dependent on environmental conditions. Individual macromolecules with specific affinity for other macromolecules self-assemble into supramolecular complexes. 1.5 Evolutionary Foundations. Nothing in biology makes sense except in the light of evolution. Theodosius Dobzhansky, the American biology teacher, March 1973. Great progress in biochemistry and molecular biology in recent decades has amply confirmed the validity of Dobzhansky's striking generalization. The remarkable similarity of metabolic pathways and gene sequences across the three domains of life argues strongly that all modern organisms are derived from a common evolutionary progenitor by a series of small changes, mutations, each of which conferred a selective advantage to some organism in some ecological niche. Changes in the hereditary instructions allow evolution. Despite the near-perfect fidelity of genetic replication, infrequent unrepaired mistakes in the DNA replication process lead to changes in the nucleotide sequence of DNA, producing a genetic mutation and changing the instructions for a cellular component. Incorrectly repaired damage to one of the DNA strands has the same effect. Mutations in the DNA handed down to offspring, that is, mutations carried in the reproductive cells, may be harmful or even lethal to the new organism or cell. They may, for example, cause the synthesis of a defective enzyme that is not able to catalyze an essential metabolic reaction. Occasionally, however, a mutation better equips an organism or cell to survive in its environment. Fig. 1-34. The mutant enzyme might have acquired a slightly different specificity, for example, so that it is now able to use some compound that the cell was previously unable to metabolize. If a population of cells were to find itself in an environment where that compound was the only or the most abundant available source of fuel, the mutant cell would have a selective advantage over the other, unmutated, wild-type cells in the population. The mutant cell and its progeny would survive and prosper in the new environment, whereas wild-type cells would starve and be eliminated. This is what Darwin meant by natural selection, what is sometimes summarized as survival of the fittest. Occasionally, a second copy of a whole gene is introduced into the chromosome as a result of defective replication of the chromosome. The second copy is superfluous, and mutations in this gene will not be deleterious. It becomes a means by which the cell may evolve, by producing a new gene with a new function while retaining the original gene in gene function. Seen in this light, the DNA molecules of modern organisms are historical documents, records of the long journey from the earliest cells to modern organisms. The historical accounts in DNA are not complete, however. In the course of evolution, many mutations must have been erased or written over. But DNA molecules are the best source of biological history that we have. The frequency of errors in DNA replication represents a balance between too many errors, which would yield non-viable daughter cells, and too few, which would prevent the genetic variation that allows survival of mutant cells in new ecological niches. Figure 1-30 which is superfluous. Several billion years of natural selection have refined cellular systems to take maximum advantage of the chemical and physical properties of available raw materials. Chance genetic mutations occurring in individuals in a population, combined with natural selection, have resulted in the evolution of the enormous variety of species we see today, each adapted to its particular ecological niche. Biomolecules first arose by chemical evolution. In our account thus far, we have passed over the first chapter of the story of evolution, the appearance of the first living cell. Apart from their occurrence in living organisms, organic compounds, including the basic biomolecules such as amino acids and carbohydrates, are found in only trace amounts in the Earth's crust, the sea, and the atmosphere. How did the first living organisms acquire their characteristic organic building blocks? According to one hypothesis, these compounds were created by the effects of powerful environmental forces: ultraviolet irradiation, lightning, or volcanic eruptions on the gases in the prebiotic Earth's atmosphere and on inorganic solutes in superheated thermal vents deep in the ocean. This hypothesis was tested in a classic experiment on the abiotic, non-biological, origin of organic biomolecules carried out in 1953 by Stanley Miller in the laboratory of Harold Urey. Miller subjected gaseous mixtures, such as those presumed to exist on the prebiotic Earth, including NH3, CH4, H2O and H2, to electrical sparks produced across a pair of electrodes, to simulate lightning, for periods of a week or more, then analyze the contents of the closed reaction vessel Fig. 1-35. The gas phase of the resulting mixture contains CO and CO2, as well as the starting materials. The water phase contained a variety of organic compounds, including some amino acids, hydroxy acids, aldehydes, and hydrogen cyanide, HCN. This experiment established the possibility of abiotic production of biomolecules in relatively short times under relatively mild conditions. When Miller's carefully stored samples were rediscovered in 2010 and examined with much more sensitive and discriminating techniques, high-performance liquid chromatography and mass spectrometry, his original observations were confirmed and greatly broadened. Previously unpublished experiments by Miller that included H2S in the gas mixture, mimicking the smoking volcanic plumes at the sea bottom, Fig. 1-36, showed the formation of 23 amino acids and 7 organosulfur compounds, as well as a large number of other simple compounds that might have served as building blocks in prebiotic evolution. Figure 1-35. More refined laboratory experiments have provided good evidence that many of the chemical components of living cells can form under these conditions. Polymers of RNA can act as catalysts in biologically significant reactions. See chapters 26 and 27, and RNA probably played a crucial role in prebiotic evolution, both as catalyst and as information repository. Ribonucleotides, the monomeric units of RNA, have not been formed in the laboratory under prebiotic conditions, so it is possible that prebiotic evolution began with an RNA-like molecule, rather than with RNA itself. Figure 1-3. RNA or related precursors may have been the first. Genes and catalysts. In modern organisms, nucleic acids encode the genetic information that specifies the structure of enzymes, and enzymes catalyze the replication and repair of nucleic acids. The mutual dependence of these two classes of biomolecules brings up the perplexing question: which came first, DNA or protein? The answer may be that they appeared about the same time, and RNA preceded them both. The discovery that RNA molecules can act as catalysts in their own formation suggests that RNA, or a similar molecule, may have been the first gene and the first catalyst. According to this scenario, Fig. 137, one of the earliest stages of biological evolution was the chance formation of an RNA molecule that could catalyze the formation of other RNA molecules of the same sequence as self-replicating, self-perpetuating RNA. The concentration of a self-replicating RNA molecule would increase exponentially, as one molecule formed several formed many, and so on. The fidelity of self-replication was presumably less than perfect, so the process would generate variants of the RNA, some of which might be even better able to self-replicate. In the competition for nucleotides, the most efficient of the self-replicating sequences would win, and less efficient replicators would fade from the population. Figure 1 to 37a. The division of function between DNA, genetic information storage, and protein, catalysis, was, according to the Rna World hypothesis, a later development. New variants of self-replicating RNA molecules developed that had the additional ability to catalyze the condensation of amino acids into peptides. Occasionally, the peptides thus formed would reinforce the self-replicating ability of the RNA, and the pair RNA molecule and helping peptide could undergo further modifications in sequence, generating increasingly efficient self-replicating systems. The remarkable discovery that in the protein synthesizing machinery of modern cells, ribosomes, RNA molecules, not proteins, catalyze the formation of peptide bonds is consistent with the Rna World hypothesis. Sometime after the evolution of this primitive protein synthesizing system, there was a further development. DNA molecules with sequences complementary to the self-replicating RNA molecules took over the function of conserving the genetic information, and RNA molecules evolved to play roles in protein synthesis. We explain in Chapter 8 why DNA is a more stable molecule than RNA, and thus a better repository of inheritable information. Proteins proved to be versatile catalysts and, over time, took over most of that function. Lipid-like compounds in the primordial mixture formed relatively impermeable layers around self-replicating collections of molecules. The concentration of proteins and nucleic acids within these lipid enclosures favored the molecular interactions required in self-replication. The Rna World Scenario is intellectually satisfying, but it leaves unanswered a vexing question: where did the nucleotides needed to make the initial RNA molecules come from? An alternative to this Rna World Scenario supposes that simple metabolic pathways evolved first, perhaps at the hot vents in the ocean floor. A set of linked chemical reactions there might have produced precursors, including nucleotides, before the advent of lipid membranes or RNA. Without more experimental evidence, neither of these hypotheses can be disproved. Biological evolution began more than 3.5 billion years ago. Earth was formed about 4.6 billion years ago, and the first evidence of life dates to more than 3.5 billion years ago. In 1996, scientists working in Greenland found chemical evidence of life, fossil molecules, from as far back as 3.85 billion years ago, forms of carbon embedded in rock that seem to have a distinctly biological origin. Somewhere on Earth during its first billion years the first simple organism arose, capable of replicating its own structure from a template, RNA, that was the first genetic material. Because the terrestrial atmosphere at the dawn of life was nearly devoid of oxygen, and because there were few microorganisms to scavenge organic compounds formed by natural processes, these compounds were relatively stable. Given this stability in eons of time, the improbable became inevitable. Lipid vesicles containing organic compounds and self-replicating RNA gave rise to the first cells, or protocells, and those protocells with the greatest capacity for self-replication became more numerous. The process of biological evolution had begun. The first cell probably used inorganic fuels. The earliest cells arose in a reducing atmosphere, there was no oxygen, and probably obtained energy from inorganic fuels such as ferrous sulfide and ferrous carbonate, both abundant on the early Earth. For example, the reaction FeS plus H2S FeS2 plus H2 yields enough energy to drive the synthesis of ATP or similar compounds. The organic compounds these early cells required may have arisen by the non-biological actions of lightning or of heat from volcanoes or thermal vents in the sea on components of the early atmosphere, CO, CO2, N2, NH3, CH4, and such like. An alternative source of organic compounds has been proposed: extraterrestrial space. Space missions in 2006, Stardust, and 2014, Philae, found particles of comet dust to contain the simple amino acid glycine and 20 other organic compounds capable of reacting to form biomolecules. Early unicellular organisms gradually acquired the ability to derive energy from compounds in their environment, and to use that energy to synthesize more of their own precursor molecules, thereby becoming less dependent on outside sources. A very significant evolutionary event was the development of pigments capable of capturing the energy of light from the sun, which could be used to reduce, or fix, CO2 to form more complex organic compounds. The original electron donor for these photosynthetic processes was probably H2S, yielding elemental sulfur or sulfate as the byproduct. Some hydrothermal vents in the sea bottom, black smokers, Fig. 1 to 36, emit significant amounts of H2, which is another possible electron donor in the metabolism of the earliest organisms. Later cells developed the enzymatic capacity to use H2O as the electron donor in photosynthetic reactions, producing O2 as waste. Cyanobacteria are the modern descendants of these early photosynthetic oxygen producers. Because the atmosphere of Earth in the earliest stages of biological evolution was nearly devoid of oxygen, the earliest cells were anaerobic. Under these conditions, chemotrophs could oxidize organic compounds to CO2 by passing electrons not to O2, but to acceptors such as, in this case yielding H2S as the product. With the rise of O2 producing photosynthetic bacteria, the atmosphere became progressively richer in oxygen, a powerful oxidant and deadly poison to anaerobes. Responding to the evolutionary pressure of what Lynn Margulis and Dorian Sagan called the oxygen holocaust, some lineages of microorganisms gave rise to aerobes that obtained energy by passing electrons from fuel molecules to oxygen. Because the transfer of electrons from organic molecules to O2 releases a great deal of energy, aerobic organisms had an energetic advantage over their anaerobic counterparts when both competed in an environment containing oxygen. This advantage translated into the predominance of aerobic organisms in O2-rich environments. Modern bacteria and archaea inhabit almost every ecological niche in the biosphere, and there are organisms capable of using virtually every type of organic compound as a source of carbon and energy. Photosynthetic microbes in both fresh and marine waters trap solar energy and use it to generate carbohydrates and all other cell constituents, which are in turn used as food by other forms of life. The process of evolution continues and, in rapidly reproducing bacterial cells, on a time scale that allows us to witness it in the laboratory. One interesting line of research into evolutionary mechanisms aims at producing a synthetic cell in the laboratory, one in which the experimenter provides every component from known, purified components. The first step in this direction involves determining the minimum number of genes necessary for life by examining the genomes of the simplest bacteria. The smallest known genome of a free-living bacterium is that of Mycoplasma. Mycoids, which comprises 1.08 megabase pairs, 1 megabase pair is a million base pairs. In 2010, scientists at the Craig Venter Institute succeeded in synthesizing the full chromosome of a mycoplasma in vitro, then incorporating that synthetic chromosome into a living bacterial cell of another species, Mycoplasma capriculum, from which the DNA had been removed, which thereby acquired the properties of M. mycoids, Fig. 1-38. This technology opens the way to producing a synthetic cell, with the bare minimum of genes essential to life. With such a cell, one could hope to study, in the laboratory, the evolutionary processes by which protocells gradually diversified and became more complex. Lynn Margulis, Figure 1-30. Eukaryotic cells evolved from simpler precursors in several stages. Starting about 1.5 billion years ago, the fossil record begins to show evidence of larger and more complex organisms, probably the earliest eukaryotic cells, Fig. 1-39. Details of the evolutionary path from non-nucleated to nucleated cells cannot be deduced from the fossil record alone, but morphological and biochemical comparisons of modern organisms have suggested a sequence of events consistent with the fossil evidence. Three major changes must have occurred. First, as cells acquired more DNA, the mechanisms required to fold it compactly into discrete complexes with specific proteins, and to divide it equally between daughter cells at cell division became more elaborate. Specialized proteins were required to stabilize folded DNA, and to pull the resulting DNA protein complexes, chromosomes apart during cell division. Second, as cells became larger, a system of intracellular membranes developed, including a double membrane surrounding the DNA. This membrane segregated the nuclear process of RNA synthesis on a DNA template from the cytoplasmic process of protein synthesis on ribosomes. Finally, according to a now widely accepted hypothesis advanced, initially, to much resistance, by Lynn Margulis, early eukaryotic cells, which were incapable of photosynthesis or aerobic metabolism, enveloped aerobic bacteria or photosynthetic bacteria to form endosymbiotic associations that eventually became permanent, Fig. 1-40. Some aerobic bacteria evolved into the mitochondria of modern eukaryotes, and some photosynthetic cyanobacteria became the plastids, such as the chloroplasts of green algae, the likely ancestors of modern plant cells. Figure 1. At some later stage of evolution, unicellular organisms found it advantageous to cluster together, thereby acquiring greater motility, efficiency, or reproductive success than their free-living single-celled competitors. Further evolution of such clustered organisms led to permanent associations among individual cells, and eventually to specialization within the colony to cellular differentiation. The advantages of cellular specialization led to the evolution of increasingly complex and highly differentiated organisms, in which some cells carried out the sensory functions, others the digestive, photosynthetic, or reproductive functions, and so forth. Many modern multicellular organisms contain hundreds of different cell types, each specialized for a function that supports the entire organism. Fundamental mechanisms that evolved early have been further refined and embellished through evolution. The same basic structures and mechanisms that underlie the beating motion of cilia in Paramecium and of flagella in Chlamydomonas are employed by the highly differentiated vertebrate sperm cell, for example. Molecular anatomy reveals evolutionary relationships. Biochemists now have an enormously rich, ever-increasing treasury of information on the molecular anatomy of cells that they can use to analyze evolutionary relationships and refine evolutionary theory. The sequence of the genome, the complete genetic endowment of an organism, has been determined for several thousand bacteria and archaea and for growing numbers of eukaryotic microorganisms, including Saccharomyces cerevisiae and Plasmodium species, plants, including Arabidopsis thaliana and rice, and animals, including Cenorhabditis elegans, a roundworm, Drosophila melanogaster, the fruit fly, mouse, rat, dog, chimpanzee, and Homo sapiens, Table 1-2. It is even possible to recover DNA samples from the tissues of extinct animals, such as Neanderthal man and woolly mammoth, and sequence it. See Chapter 8. With such sequences in hand, detailed and quantitative comparisons among species can provide deep insight into the evolutionary process. Thus far, the molecular phylogeny derived from gene sequences is consistent with, but in many cases more precise than, the classical phylogeny based on macroscopic structures. Although organisms have continuously diverged at the level of gross anatomy, at the molecular level the basic unity of life is readily apparent, molecular structures and mechanisms are remarkably similar from the simplest to the most complex organisms. These similarities are most easily seen at the level of sequences, either the DNA sequences that encode proteins or the protein sequences themselves. Figure 1 to 40 Evolution. Table 1 to 2 A few whose genomes complete organ helic influence Synechocystis Bacillus sub Arabidopsis Drosophila Muse Homos Paris. Sources. When two genes share readily detectable sequence similarities, nucleotide sequence in DNA or amino acid sequence in the proteins they encode, their sequences are said to be homologous, and the proteins they encode are homologs. If two homologous genes occur in the same species, they are said to be paralogous, and their protein products are paralogs. Paralogous genes are presumed to have been derived by gene duplication, followed by gradual changes in the sequences of both copies. Typically, paralogous proteins are similar not only in sequence, but also in three-dimensional structure, although they commonly have acquired different functions during their evolution. Two homologous genes, or proteins, found in different species are said to be orthologous, and their protein products are orthologs. Orthologs usually have the same function in both organisms, and when a newly sequenced gene in one species is found to be strongly orthologous with a gene in another, this gene is presumed to encode a protein with the same function in both species. By this means, the function of gene products, proteins, or RNA molecules, can be deduced from the genomic sequence without any biochemical characterization of the molecules themselves. An annotated genome includes, in addition to the DNA sequence itself, a description of the likely function of each gene product, deduced from comparisons with other genomic sequences and established protein functions. Sometimes, by identifying the pathways, sets of enzymes, encoded in a genome, we can deduce from the genomic sequence alone the organism's metabolic capabilities. The sequence differences between homologous genes may be taken as a rough measure of the degree to which the two species have diverged during evolution of how long ago their common evolutionary precursor gave rise to two lines with different evolutionary fates. The larger the number of sequence differences, the earlier the divergence in evolutionary history. 1. Can construct a phylogeny, family tree, in which the evolutionary distance between any two species is represented by their proximity on the tree. Fig. 1.5 is an example. In the course of evolution, new structures, processes, or regulatory mechanisms are required, reflections of the changing genomes of the evolving organisms. The genome of a simple eukaryote such as yeast should have genes related to formation of the nuclear membrane, genes not present in bacteria or archaea. The genome of an insect should contain genes that encode proteins involved in specifying a characteristic segmented body plan, genes not present in yeast. The genomes of all vertebrate animals should share genes that specify the development of a spinal column, and those of mammals should have unique genes necessary for the development of the placenta, a characteristic of mammals, and so on. Comparisons of the whole genomes of species in each phylum are leading to the identification of genes critical to fundamental evolutionary changes in body plan and development. Functional genomics shows the allocations of genes to specific cellular processes. When the sequence of a genome is fully determined and each gene is assigned a function, molecular geneticists can group genes according to the processes: DNA synthesis, protein synthesis, generation of ATP, and so forth, in which they function, and thus find what fraction of the genome is allocated to each of a cell's activities. The largest category of genes in E. coli, A. Thaliana, and H. sapiens consists of those of, as yet, unknown function, which make up more than 40% of the genes in each species. The genes encoding the transporters that move ions and small molecules across plasma membranes make up a significant proportion of the genes in all three species, more in the bacterium and plant than in the mammal, 10% of the 400 genes of E. coli, 8% of the 27,000 genes of A. Thaliana, and 4% of the 20,000 genes of H. sapiens. Genes that encode the proteins and RNA required for protein synthesis make up 3% to 4% of the E. coli genome, but in the more complex cells of A. Thaliana, more genes are needed for targeting proteins to their final location in the cell than are needed to synthesize those proteins, about 6% and 2% of the genome, respectively. In general, the more complex the organism, the greater the proportion of its genome that encodes genes involved in the regulation of cellular processes, and the smaller the proportion dedicated to basic processes, or housekeeping functions, such as ATP generation and protein synthesis. The housekeeping genes typically are expressed under all conditions, and are not subject to much regulation. Genomic comparisons have increasing importance in human biology and medicine. The genomes of chimpanzees and humans are 99.9% identical, yet the differences between the two species are vast. The relatively few differences in genetic endowment must explain the possession of language by humans, the extraordinary athleticism of chimpanzees, and myriad other differences. Genomic comparison is allowing researchers to identify candidate genes linked to divergences in the developmental programs of humans and the other primates, and to the emergence of complex functions such as language. The picture will become clearer only as more primate genomes become available for comparison with the human genome. Similarly, the differences in genetic endowment among humans are vanishingly small compared with the differences between humans and chimpanzees, yet these differences account for human variety, including differences in health and in susceptibility to chronic diseases. We have much to learn about the variability in genomic sequence among humans and the availability of genomic information will almost certainly transform medical diagnosis and treatment. Several monumental studies in which the entire genomic sequence has been determined for hundreds or thousands of people with cancer, type 2 diabetes, schizophrenia, and other diseases or conditions have allowed the identification of many genes in which mutations correlate with the medical condition. Each of those genes codes for a protein that, in principle, might become the target for drugs to treat that condition. We may expect that for some genetic diseases, palliatives will be replaced by cures, and that for disease susceptibilities associated with particular genetic markers, forewarning and perhaps increased preventive measures will prevail. Today's medical history may be replaced by a medical forecast. Summary 1.5 Evolutionary Foundations. Occasional inheritable mutations yield organisms that are better suited for survival and reproduction in an ecological niche, and their progeny come to dominate the population in that niche. This process of mutation and selection is the basis for the Darwinian evolution that led from the first cell to all modern organisms. The large number of genes shared by all living organisms explains organisms' fundamental similarities. Life originated about 3.5 billion years ago, most likely with the formation of a membrane-enclosed compartment containing a self-replicating RNA molecule. The components for the first cell may have been produced near thermal vents at the bottom of the sea or by the action of lightning and high temperature on simple atmospheric molecules such as CO2 and NH3. The catalytic and genetic roles played by the early RNA genome were, over time, taken over by proteins and DNA, respectively. Eukaryotic cells acquired the capacity for photosynthesis and oxidative phosphorylation from endosymbiotic bacteria. In multicellular organisms, differentiated cell types specialize in one or more of the functions essential to the organism's survival. Knowledge of the complete genomic nucleotide sequences of organisms from different branches of the phylogenetic tree provides insights into evolution and offers great opportunities in human medicine. Key terms: